Let me ask you this question. Do you have any transcription factors in your body? I sure hope the answer is yes, because otherwise you are going to have hard time keeping your cells running. So what are these transcription factors? Transcription factors are proteins that regulate the transcription of genes. That is, they're copying into RNA on the way of making a protein. The human body contains many transcription factors, so does the body of a bird, tree, or fungus. Transcription factors help ensure that the right genes are expressed in the right cells of the body at the right time. So, the key control point is that transcription is the process where a gene's DNA sequence is copied or transcribed into an RNA molecule. Transcription is a key step in using information from a gene to make a protein. If you're not familiar with those ideas, please just watch the central dogma video for a solid introduction. Gene expression is when a gene in DNA is turned on, that is used to make the protein it specifies. Not all the genes in your body are turned on at the same time or in the same cells or part of the body. For many genes, transcription is the key on-off control point. If a gene is not transcribed in a cell, it cannot be used to make a protein in that cell. If a gene does get transcribed, it's likely going to be used to make a protein or expressed. In general, the more a gene is transcribed, the more protein that will be made. Various factors control how much a gene is transcribed. For instance, how tightly the DNA of the gene is wound around its supporting proteins to form chromatin can affect a gene's availability for transcription. Proteins called transcription factors, however, play a particularly central role in regulating transcription. These important proteins help determine which genes are active in each cell of your body. So, what has to happen for a gene to be transcribed? Let's ask this question: that what has to happen a gene to be transcribed? The enzyme RNA polymerase, which makes a new RNA molecule from a DNA template, must attach to the DNA of the gene. It attaches at a spot called the promoter. In bacteria, RNA polymerase attaches right to the DNA of the promoter. You can see how this process works and how it can be regulated by transcription factors in the lac operon or TRP operon videos. In humans and other eukaryotes, there is an extra step. RNA polymerase can attach to the promoter only with the help of proteins called basal general transcription factors. They are part of the cell's core transcription toolkit needed for the transcription of any gene. However, many transcription factors, including some of the coolest ones, are not the general kind. Instead, there is a large class of transcription factors that control the expression of specific individual genes. For instance, a transcription factor might activate only a set of genes needed in certain neurons. So let's get to the point that how do these transcription factors work. A typical transcription factor binds to DNA at a certain target sequence. Once it's bound, the transcription factor makes it either harder or easier for RNA polymerase to bind to the promoter of the gene. Activators, some transcription factors activate transcription. For instance, they may help the general transcription factors and or RNA polymerase bind to the promoter, as shown in this diagram, you see. And other transcription factors repress transcription. This repression can work in a variety of ways. As one example, a repressor may get in the way of the basal transcription factors or RNA polymerase, making it so they cannot bind to the promoter or begin transcription. The binding sites for transcription factors are often close to the gene's promoter. However, they can also be found in other parts of the DNA, sometimes very far away from the promoter, and still affect transcription of the gene. The flexibility of DNA is what allows transcription factors at distance binding sites to do their job. The DNA looks like cooked spaghetti to bring far off binding sites and transcription factors close to the general transcription factors or mediator proteins. So in this picture, an activating transcription factor bound at a far away site helps RNA polymerase bind to the promoter and start transcribing. How is this different from E. coli? So far, human and other eukaryotic transcription factors do not seem all that different from the transcription factors we have seen in bacteria. They bind DNA and make it easier or harder for RNA polymerase to do its job, just like the lac repressor protein of E. coli. In general, this is a pretty good takeaway. Proteins that control transcription tend to act in similar ways, whether they are in your own cells or in bacteria that live in your nose. The main differences are mechanical, how far away regulatory sites are, whether basal transcription factors are needed, etc. However, there are also some meaningful differences in how transcription factors are used in humans. Humans and other eukaryotes are complex. We are made up of trillions of cells organized into unique tissues and body structures. Each cell in your body must run its own program of gene expression. So turning genes on in specific body parts, some genes need to be expressed in more than one body part of the or type of the cell. For instance, suppose a gene is needed to be turned on in your spine, skull, and fingertips, but not in the rest of your body. How can transcription factors make this pattern happen? A gene with this type of pattern may have several enhancers, faraway clusters of binding sites for activators, or silencers, the same thing for repressors. Each enhancer or silencer may activate or repress the gene in a certain cell type of or body part, binding transcription factors that are made in that part of the body. Okay, so an example, a modular mouse. As an example, let's consider a gene found in mice called TBX4. this gene is important for the development of many different parts of the mouse body, including the blood vessels and the hind legs. During development, several well-defined enhancers drive TBX4 expression in different parts of the mouse embryo. The diagram in here shows some of the TBX4 enhancers, each labeled with the body part where it produces expression. Enhancers like those of the TBX4 gene are called tissue-specific enhancers. They control a gene's expression in a certain part of the body. Mutations of tissue-specific enhancers and silencers may play a key role in the evolution of body form. How could that work? Suppose that a mutation or change in DNA happened in the coding sequence of the TBX4 gene. The mutation would inactivate the gene everywhere in the body, and a mouse without a normal copy would likely die. However, a mutation in enhancer might just change the expression pattern a bit, leading to a new feature with a shorter leg without killing the mouse. Okay, so can cells do logic? This is the next question. Not in the same way as your amazing brain, of course. However, cells can detect information and combine it to determine the correct response in much the same way that your calculator detects push buttons and outputs an answer. We can see an example of this molecular logic when we consider how transcription factors regulate genes. Many genes are controlled by several different transcription factors with a specific combination needed to turn the gene on. This is particularly true in eukaryotes and is sometimes called combinatorial regulation. For instance, a gene may be expressed only if activators A and B are present and if repressor C is absent. Excuse me. The use of multiple transcription factors to regulate a gene means that different sources of information can be integrated into a single outcome. For instance, imagine that activator is present only in skin cells. Activator B is active only in cells receiving divide now signals, growth factors from neighbors. Repressor C is produced when a cell's DNA is damaged. In this case, the gene would be turned on only in skin cells that are receiving division signals and must undamaged healthy DNA. This pattern of regulation might make sense for a gene involved in cell division in skin cells. In fact, the loss of proteins similar to repressor C can lead to cancer. Real life combinatorial regulation can be a bit more complicated than this. For instance, many different transcription factors may be involved, or it may matter exactly how many molecules of a given transcription factor are bound to DNA. Okay, so let's talk about the key points of this video presentation. So transcription factors are proteins that help turn specific genes on or off by binding to nearby DNA. Transcription factors that are activators boost a gene's transcription. Repressors decrease the transcription. Groups of transcription factors, factor binding sites are called enhancers and silencers can turn a gene on and off in specific parts of the body. Transcription factors allow cells to perform logic operations and combine different sources of information to decide whether to express a gene.